DFG Science TV. Search for a cure. The fight against forgetting. Zebrafish help understand Alzheimer's.
Doctoral researcher Dominik has taken a big step closer to the goal of developing a treatment for Alzheimer's disease. He's managed to implant a human Alzheimer's gene into zebrafish. But first, he has to find out how the fish will react to the new gene and the disease. What would be incredibly important now is for us to see symptoms in these fish. So, in the same way as in a patient, we need to see deposits forming or nerve cells dying out. Because only if we find these symptoms can we really use the fish to also test drugs on, for example, and to find new treatments. Whether the fish show symptoms of Alzheimer's, however, Dominik can only assess by examining their cells. Affected cells have agglutinations of the tau protein. The agglutinations, however, only become visible by using a special staining method, so-called silver staining. This is no easy task, as before he can stain the fish, he must freeze them and cut them into extremely thin slices with a device known as a cryostat. This allows the cells to better absorb the colouring. The preparations are then immersed into many different dye solutions and rinsed out. This guarantees that the colouring only sticks to the agglutinations. And it's actually worked. Agglutinations can be detected in 5-week-old fish. A first success. But to find out whether the cells in the fish actually die out, Dominik can only test on a live fish by means of a special dye that is added to the water. This is absorbed only by dyeing cells, and it then colours them yellow. By doing so, it's possible to observe, using a microscope, how the nerve cells die out on a live creature. Yes, I've now just put the Alzheimer's fish under our modern layer scanning microscope for the first time. This has a very high resolution, so you can really look very closely and study the nerve cells in detail. And so I was able to observe live for the first time, over an extended period, how the nerve cells died out. And that's something which we think happens in the patient in a very similar way, that the cells simply die out. Dominik has done it. He's developed a new animal model that shows typical symptoms of Alzheimer's disease. We can now draw inferences from the fish to the patient and develop ideas as to how we can prevent this dying off of the cells. Will Frauke manage to find a drug that delays the death of the cells?
Visit DFG Science TV for more information. Awaken the researcher within you.